Hello, hello students. This is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning sessions in immunology. Today, in this session, we learn about vaccination. Vaccination is an effective strategy for restraining infections. Vaccines are those that are successful in provoking high affinity antibodies and memory lymphocyte cells. Most of the vaccine developed function by inducing humoral immune response in the host. Vaccination is the process of transferring or injecting the vaccine, that is, the part of the pathogen, or it is the attenuated strain of the antigen, will be injected into the host organism by mixing that with the carrier substance. It may be adjuvant. So, vaccination is the process of injecting the vaccine into the host organism. Vaccination is an effective strategy for restraining infections. There are different types of vaccines. So these vaccines are classified like this. First, attenuated and inactivated bacterial and viral vaccines. Vaccines containing intact non-pathogenic microbes are made after attenuating the virus or by killing the microbes, taking care of their immunogenicity. Attenuated viral vaccines prove beneficial because they evoke effective innate and adaptive immune response. Live attenuated bacterial vaccines used nowadays, which offers protection but for small duration, while live attenuated viral vaccine elicit good response and long lasting immunity. Viral vaccines perform better because of their adoption to the cell's culture. However, live viral vaccines always face a potent risk of reversion to virulence, and hence, safety is the main concern for such viruses. To minimize such risk, inactivated vaccines are used, such as influenza vaccine. Next one is the purified antigen or the subunit vaccines. As the name suggests, these subunit vaccines are those that contain a purified antigen and also needs to be given along with an immunogenic enhancer called an adjuvant. The construction of subunit vaccine involves isolation of specific protein or a molecule from a virus or bacteria before administration. Examples include diphtheria and tetanus are the best examples of the subunit vaccines. Next we will move on to the another vaccine, that is the synthetic antigen vaccines. This concept involves the identification of an epitope or an antigenic determinant and designing it in the laboratory to be used in the future as vaccine. Next is live viral vaccines. Live viral vaccines involving recombinant viruses. Involving recombinant viruses. Recombinant viruses involve the insertion of genes that encode an antigen into a non cytopathic virus, which provide immunity following its introduction to susceptible host. They offer protection by eliciting both innate and adaptive immune response. However, in some cases, safety is the issue. Next, we'll move on to the another type of the vaccine, that is DNA vaccines. As the name suggests, this forms the basis of the most fundamental work being done in the current times. DNA vaccine strategy involves inoculation of a plasmid containing complementary DNA encoding a protein antigen. The DNA vaccines elicit both humoral and cell mediated response, even without any adjuvant administration. But their effectiveness needs more experimentation and verification. So, this is regarding the types of vaccines. Next, we'll move on to the mechanism which occur inside the host organism after it has been injected into the uh, host organism, or when it is administered into the individual. So the what will happen inside the immune system? For example, first antigen X. So individual is exposed to antigen X. From this is the time that is represented in days on x-axis, and this is the antibody level, also called as the antibody titer. As soon as the individual is infected by the antigen X, immediately there will not be the production of the antibody. It will take few days, or the few days will be taken to synthesize the antibody. And now, soon after it recognizes, it activates the clone of cells and it starts secreting the antibodies. Initially, IgM antibodies will be produced, and these IgM antibodies, they form both effector cells as well as the plasma cells, or the memory cells will be formed, and the effector cells clear off the infection at that moment, whereas the memory lymphocyte retain the memory for the further subsequent infection. Then if the same individual is infected by the antigen X after the first administration, so the amount of antibody level in the host organism increases. So first administration, next we will be going for the booster dose. So here there will be increase in the amount, or the titer of the IgM antibody. So this is regarding the primary response. And after that, if the same individual, naturally when he is infected by the same antigen, the immunological memory which is there in the immunity will trigger the antibody titer, and the IgG antibody concentration will be increased, and heightened immune reaction will be raised against the antigen X and will inhibit its activity, and resistance will be exhibited by the host organism. So it has both primary response and the secondary response. So vaccination, so initially when we administer the vaccine, IgM antibodies are produced, and later there will be secondary response when the individual further frequently when he infected by the antigen X, IgG antibodies will be produced by the memory B cells or the memory lymphocytes, and they inhibit the onset of infection inside the host organism. Next is, so this is the days after injecting the vaccine, and how the antibody level will be increased inside the host organism. So when you inject the vaccine, administer the vaccine into the host organism, in a pool of lymphocyte, the specific navy B cells will be activated, and they proliferate and they produces plasma cells or the effector cells, and they secrete the IgM antibodies, and this group of cell will clear off the infection, and these antibodies in the primary response will be of low affinity antibodies, and the portion or the fraction of the activated B lymphocytes also forms the memory lymphocyte, and these memory cells will be there in the host individual who has been injected or administered with the antigen or the vaccine. Later, when you give the booster doses of the same the vaccine, there will be increased production of the antibodies, especially IgG antibodies, and immediately the infection will be, or the immunity will be triggered to produce more and more memory B lymphocyte. So here you have the primary antibody response and the secondary antibody response, that is due to the over a period of time, the immune system recognizes the vaccine and it starts activating the B lymphocytes to produce the IgM antibody later that leads to the production of the IgG antibodies. So this mechanism takes place in the when we administer the vaccine in the host organism. When you see the graph of the primary response, we will find the variation wherein, so this phase is in case of the primary response, an individual first encounter with the particular immunogen called priming event and leads to a relatively weak short-lived response designated as the primary immune response. This is divisible into several phases. In the humoral branch of immune system, antigen induces a clonal proliferation of the B lymphocyte. It leads to clonal proliferation of the B lymphocytes into antibody secreting plasma cells and memory cells. So here you will find the different phases. First is lag phase. Lag or latent phase is the time between the initial exposure of an immunogen and the detection of antibodies in the circulation. So this average about 1 week in the humans. During this period, activation of Th cells and the B cells takes place. Next comes the exponential phase. In the exponential phase, there is rapid increase in the quantity of the antibodies reflects the increased number of secretory cells or the effector cells. Then comes the stationary phase. In the stationary phase, we'll find the plateau level wherein the antibody level remains relatively constant because secretion and the degradation are occurring simultaneously in equal proportion, and hence steady state or the plateau level will be observed. Following which the secretory cells, the degradation rate increases and hence we'll find the decline in the antibody concentration, that is called the decline phase. So that is regarding the primary antibody response. Next is the secondary antibody response. The secondary antibody response reflects the activity of the clonally expanded population of memory B cells. So actually the antibodies are produced by the memory B cells. They are involved in the secondary antibody response. These memory cells respond to the antigen more rapidly than the naive B cells. In addition, because there are many more memory cells, they were navy B cells for the primary response. More plasma cells are generated in the secondary response and the antibody levels are consequently increased. Now, when you compare the primary antibody response and the secondary antibody response, maybe around 100 to 1000 fold it will be increased. Subsequent encounter with the same immunogen lead to the responses that are qualitatively similar to the primary response, but it activates marked quantitative differences in the antibody level, and that's why you'll see the difference in the antibody concentration both in the primary response and the secondary response. So this is in case of the secondary response. This is in case of the antibody is produced in the primary response. The amplified population of the memory cells accounts for the rapidity and the intensity that distinguishes a secondary response from the primary response. So this is regarding the vaccination. So that we come across in our life, day-to-day life, to bring or to activate the immune system against various infection. So in this session, we learned what is vaccination, what are vaccines and different types of vaccines, and the mechanism involved in vaccination, like primary antibody response and the secondary antibody response, and we learned the differences between the IgG antibodies in secondary response and the IgM antibody response. I hope you understood today's topic. Thank you.